Hello, Life Science. Hope you're doing well. Today, we're going to do a quick revision of the main ideas of Chapter 6, and we're going to correct the homework. We have seen in this chapter that white blood cells are the effectors of immunity. These cells are the monocytes, the granulocytes, and the lymphocytes. The most important thing is to know how an antigen is recognized by the immune system, so an immune response can be triggered. The recognition of the antigen is different between lymphocyte B and lymphocyte T. Look at the figure, please. Lymphocyte B recognizes the antigen directly. How? Through the antibody, which is the membrane receptors of lymphocyte B. However, look at LT. The lymphocyte T cannot bind and recognize the antigen directly. However, this recognition should be done through a cell that we call an antigen-presenting cell. This antigen is processed inside the cell, and the resulting peptide fragment is displayed on the surface of the cell, of course on MHC or HLA. And thus here T lymphocyte makes a double recognition of the complex MHC and the peptide. I chose for you this caricature to show you how a T cell recognizes the antigen. The T cell, you can see it. It is asking, what is this? So it's unable to recognize the antigen in front of it. You see the microbes? However, when the macrophage suggests it helps, the T cell then recognizes the antigen carried by the macrophage, and you see how this T cell is happy. Eureka, it's a peptide. Okay. The recognition of the antigen by T lymphocytes, it differs between T4 and T8. Let us talk first about T8. In a first step, there is antigen association. So the antigen is going to be associated to MHC class 1 in the case of cell infected by this antigen. Then in a second step, you see the T8, it recognizes the antigen carried by MHC class 1 molecule on the surface of the self-infected cell. Or it could be a tumor cell, not necessarily an infected cell. Let us now talk about the T4. In the first step, the antigen binds to MHC class 2 molecule found on the surface of the APC, that could be the macrophage. Then in a second step, the T4 recognizes the antigen carried by MHC class 2 on the surface of this APC. So in both cases, you see that T8 and T4, they recognize antigens carried by MHC or HLA. T8 recognizes the antigen carried by HLA class 1. T4 recognizes the antigen carried by HLA class 2. Okay, we can start now with the correction of the exercises. Let's start with exercise 2. The first question, A, complete the above table, checking with T for accepted blood transfusions and with X for rejected transfusions. Okay, you have the table above, and it is filled with the answers for a true or X. Okay, so for the donor being O negative and the recipient being O negative, you see that transfusion can be done. And the same for O positive, For A negative, A positive, then O negative can donate blood to all the blood groups. O positive, it donates also to all blood groups, only that are positive and cannot donate blood to those who are negative. What about A negative? A negative can give blood only to A and AB, negative and positive. A positive can donate blood only to A and AB positive. B negative can donate blood to B and AB, whether negative or positive. B positive donates blood only to B and AB positive. AB negative, as you can see, cannot donate blood to any of the other blood groups except to itself, AB, negative and positive, and AB positive donates blood only to AB positive. In the second question, referring to the table, specify what blood group is a universal donor and what blood group is a universal recipient. Okay? Of course, no need here to add justify at the end because you know that specify means justify the answer. So universal donor is O negative. Why? Because as you see in the table, it can donate blood to all blood groups. It's not rejected by any other blood group. And the main reason for this is the absence of antigens. It doesn't have antigens. While AB positive is the universal recipient, because as you see in the table, okay, in the last column, it can receive blood from all other blood groups. So it doesn't reject any other group. So it's a universal recipient, mainly because it doesn't have antibodies, anti-A and anti-B. Last question. Indicate which molecules of the donor and the recipient are involved in the rejection of a blood transfusion. So you know that this is the main condition for blood transfusion. It's the donor's antigens or agglutinogens and the recipient agglutinins or antibodies that are involved in transfusion rejection. Of course, the donor's antigen and these agglutinins of the recipient must be different, otherwise they will agglutinate together. Exercise 3. The next document sums up the results of the percentage of graft survival in man during 5 years. So you see in the graph, we have here the variation of the percentage of graft survival, okay? Of course, as a function of time, but also as a function of the relation between donor and recipient. And this relation is indicated in the document. It could be case of the blue curve number 1, brothers and sisters of identical MHC, green parents having 50% of MHC genes in common, and in orange non-related individuals, which means 0% in common MHC genes between donor and recipient. And the star means that the treatment is done without immunosuppressors, especially when someone receives a graft. Usually this individual is provided with immunosuppressors that are medicines that decrease the immunity so that this individual tolerates the graft and doesn't reject it after a certain period of time. So here in the case of graft between non-related individuals, there was no treatment with immunosuppressors. So you see there is rejection. Okay, you are asked to first analyze the results obtained in the above graph and second to draw out a conclusion. Analysis. Of course, in order to do the analysis, you should know that you should indicate clearly the variable factor here in this experiment. So what's the variable factor here? Yes, it's the relation donor and recipient, whether the similarity of MHC is 100% blue or 50% green or 0% orange. So how would you analyze? When the donor is a brother or a sister of identical MHC, first curve, the percentage of graft survival remains 100% for 5 years. While when the donor is one of the parents having 50% of MHC genes in common, the percentage of survival decreases to 75% during the first 2 years and remains constant till the 5th year. On the contrary, when the donor and the recipient are not relatives, the percentage of graft survival decreases to 10% and is rejected during the first year. So, this is the analysis of the graph. You see, always the variable is indicated when the donor is a brother or a sister of identical MHC, or when the donor is having 50% of MHC in common, etc. Okay, what is the conclusion? The survival of the graft depends on the degree of HLA compatibility between donor and recipient. So, the more this compatibility, the more is the survival of the graft. Exercise 4. In 1974, Zinker-Nagel demonstrated the double recognition of peptide HLA complexes by TCR based on the following experiment. Mice of strain X were infected with CML virus, choriomeningitic leukemia virus, that causes a serious but non-fatal cerebral disease. 1 week later, T cells from the infected mice were collected and put in culture in the presence of infected or non-infected mouse cells derived either from one strain X or from a different strain referred to as Y strain. The following results were obtained, and the question is interpret the above results. To facilitate the understanding of the experiment, I schematized it as you can see. So the mouse of strain X was previously infected with CML, then 1 week later T lymphocytes are taken and added to the 4 different cultures you see in front of you. Okay, how are you going to interpret these experiments? Let's look for the variable factors. If you look at experiments 1 and 2, you see that in both cultures, the cells belong to strain X. So this is the constant factor. But those of culture 1 are infected and those of culture 2 are not infected. So here the variable factor is the infection with CML virus. If you look at experiment 1 and 3, you see that both cultures contain infected cells with CML. But in culture 1, the cells are of strain X, while in culture 3, they are of strain Y. So this is another variable factor, which is the strain. So we should focus on these 2 variable factors, which is the infection with the same CML virus and the strain, whether it's the same or a different strain. So let's see. How is the answer going to be? T lymphocytes of mice of strain X destroy the infected cells of the same strain X. Case 1. You see lysis means destruction of these cells. So T lymphocytes have destroyed these cells. but they don't lyse or destroy the non-infected X cells, case 2, nor the Y cells, case 3, of different strain, whether infected in case 3 or not infected, case 4. So this is the analysis. What would be the significance? This shows that T lymphocytes originating from mice of strain X destroy only the cells of the same strain X. So you saw that Y strain are not destroyed and that are infected by the same CML virus. So, and this is what we call the double recognition. So T lymphocyte should recognize the cells that belong to the same strain and that are infected by the same virus. Last exercise, exercise 8. We can separate the proteins of the blood serum by electrophoresis. Document 1 represents the curves relative to the electrophoresis of the serum of rabbit A0 and that of rabbit A1. The peak of albumin is identical in both cases. A0 is a control rabbit, A1 is a rabbit injected with tetanus toxoid 15 days ago. The question is, interpret the obtained results. Okay. If you look at document 1, you see we have curves in different colors. In red corresponds to the measurements of certain antibodies found in the serum of rabbit A0, that is the control rabbit. In green, these are the measurements of certain antibodies found in the serum of rabbit A1 that actually is injected with tetanus toxoid 2 weeks ago. And of course, there is, other than the globulins that are the antibodies, there is measurement of a protein called albumin found in the serum. Okay, what is tetanus toxoid? Actually, it is an attenuated toxin produced by the tetanus bacteria. So, and this is what we call vaccine, and we're going to take this notion later on in Chapter 7. Now, the question is to interpret these results. So, of course, anytime you are asked to analyze or to interpret, you should look for the variable factor. What is the variable factor here? It's clear in the given that whether the rabbit is injected or not with the antigen, the tetanus toxoid, that is a weakened antigen, it is chosen to be weak, so it doesn't cause the death of the rabbit. So the tetanus toxoid here represents the antigen. So the antigen, if it infects the rabbit or not, this is the variable factor. So let's analyze. The plasma of rabbit A0 and A1 have the same amount of albumin proteins, of alpha globulins, and almost also of beta globulins. But rabbit A1, which have received the tetanus toxoid, contains in the serum a higher amount of gamma globulin proteins than that of rabbit A0. So you see, I related the variable to the result. The rabbit A1 that received the injection of the antigen shows a higher amount of gamma globulin. This indicates that the contact of the rabbit with the antigen tetanus toxoid stimulates the secretion of high amounts of gamma globulin proteins or of the antibodies. And of course, these antibodies are the antibodies directed against the tetanus toxoid. Let's continue. In rabbit A1, at the level of the nodes neighboring the site of inoculation of the tetanus toxoid, we find a large number of cells represented in document 2. Cell X is transformed into cell Y. Name the structures from 1 to 10 as well as cells X and Y without reproducing them. So, if you look at the figures, cell X and cell Y, you see that cell Y shows a bigger cytoplasm with a bigger number of organelles like the mitochondria, the endoplasmic reticulum. So, let's first label the structures numbered from 1 to 10. So 1 is the cytoplasm, 2, plasma membrane, 3 is the nucleus. For the second cell Y, 4, this is the rough or granulated endoplasmic reticulum. 5 is the nucleus, 6 mitochondria, 7 is the cytoplasm, and 8 are the Golgi bodies. 9, the Golgi vesicles or secretory vesicles, and 10 is the plasma membrane. Indicate the main function, or first, what is cell X? Cell X is LB, and cell Y is the plasma cell, and this is the lymphocyte B that transformed into plasma cell in order to be able to secrete the antibodies specific for the antigen, the tetanus toxoid antigen. So you know that antibodies are proteins, and the synthesis of proteins, it requires the presence of a huge number of organelles involved in the synthesis, like the mitochondria that provides energy, Golgi bodies that store also this protein, the endoplasmic reticulum that carry the ribosomes that are the main site of protein synthesis. So part C, what is the main function of cell Y? Then the main function of cell Y would be the production of antibodies. Thank you for your attention. Prepare your questions and see you soon.